Hello, welcome to part 2 of this series where I explain how to use the ImageJ Fiji plugin for semi-automatic cell-by-cell object-based colocalization analysis. In part 1 I showed you how to generate images that are more optimized for counting cells, and in this part 2 I will explain how to count the cells, how to designate which markers cells are positive and negative for, and how we can export such colocalization data into Excel and then visualize the combined data in 3D using a Matlab script, which I have described and which I provide in this paper here. So please go online and search for this title and you will find the paper. And it is free and open for everyone on this site, Scientific Reports. The name of this plugin that we will look at in part 2 to count cells is Colocalization Object Counter. And I will now quickly show how to install the plugin. All the instructions and details can be found online here in this original paper. Just search for the title. So, first to install, go to Download and Installation, and then simply download Fiji, and then open Fiji, and go to Help, and then Update, and wait a little bit until it updates. And then this update menu comes in, and here we have to add a new plugin, and this is done by clicking Manage Update Sites and then click Add Update Site, and then you have to add this URL here and be very careful that you don't have any spaces in the end or in the beginning. Here it was a space that I had to delete, and then you make sure this is activated and you click Close here and Apply Changes. For me it is not showing because I have already installed this plugin. But then you click Close and it will automatically install. And now after you restart Fiji, it should show up here under plugins and under Colocalization Object Counter. So in this video I recommend you to press yes whenever this enable hotkeys pops out. In this video we will look at how we can count the cells in the image that we created from the previous part with the other plugin, the Colocalization Image Creator. But you can use this tool to count any kind of images that you have, and this is just an example with my images. So I suggest you just try it yourself, download the plugin and see if it works for you. So let's have a quick overview of how this plugin works and what it can do. So first of all, here I have opened the image that we created in the previous plugin, in the previous video, but it is not very important how you create your image, but it works very nicely together with the first plugin, especially because of these outlines that are created, that the plugin has this one setting for detecting what's inside these outlines. But otherwise you can use any kind of image. Most of the functions work with any kind of image. First thing you should do is make sure that you save this image. If you work with an image that is not saved, you would get an error or some other kind of error that you just have to save it. I will explain later why. So in general, when you start this plugin, the first thing it does is to activate the multipoint tool, and what you can then do is to just start and marking all of your cells. And this will be the basis for your cell counts. You can also do it semi-automatically by putting in some detection settings here and clicking Find 2D Maxima, and then it will try to guess some of the cells, and you can then inspect and you can make changes, you can move and you can delete and you can add new cells if you want. So that's the semi-automatic nature of this plugin. You can also look at the other channels. And then when you have detected some cells, you click this green button, and this will convert all the multipoints to overlays. And this looks like this. And then you can clear the multipoints again. We delete them. So these overlays, they are the actual cells, and this is what you now have designated as cells. And here you can change which type of overlay so that you want to activate, so that you can tell the software that these cells here or these in this channel, they are of category 3, and if you do it like this, now you have some different designations here. So in the top area here is where you work with these categories, so you can quantify different types of signals that you see in different cells. Here you have some options for how to visualize different things. You can set the color, you can set the sizes, and some other tools that we will explain later. You can save the counts and you can load counts, and here is the automatic detection parameters. And then finally you have the clear multipoints button here, which just clears all these multipoints, and you can use this one to toggle multipoints that are inside or outside other cells. We will see later why this is important. And you can also click to make multipoints in all the cells that you have, and then you can for example use this to designate all of them with a different category, like so. Now all of these also became category 8 except for these 3. So let's clear the multipoints again, and let's look now a bit more in detail on this plugin and what it can. So before you actually use this plugin, you have to save the picture, because all the data will be saved. Let me just locate this folder first. Where did we put, let me make a new folder, demo, and I call it output image, because all the data, the counting data, will be automatically saved in this same folder. So you see here, I will keep this up here, and you can see that a lot of files are automatically created in this folder here as we try to process this image. So just to demonstrate first, now this counts was automatically created, this folder. So all your data will be in the same folder as your original image. And the way that this works is described in this figure in the paper here. That let me just check, am I still recording? Yes. So you have 3 modes here. You have add category, set category, and delete cell, and its default is add category. And the way it works is that on any image, like we had this output image, you mark some cells that you believe are part of your set of cells. This can be also done automatically with this automatic detection we will look at later. And then after you mark them, you click this green button when you are sure that these are actual cells, and then this will be, I will now clear the multipoints, delete, and now this represents your actual ID of your cell data. So you are claiming here that we have 10 cells, and these are all category 1. That means that they have no signals, and you could add more, and you could click the green button, and let's clear it again, and you see now we have 14 cells with the specific X, Y, and Z location. And you could now you can say, oh, now I want to count those that have marker number 2 positive. So for example, if this cell, I was a bit inaccurate with this location, but if we say this cell is also positive for number 2 signal, then you add a cross inside of this circle, and the same here inside of the circle, and you have enabled this, and now you click convert multipoints to overlays using current mode, which is add category. So we are now adding number 2 to this, and now you see that these cells are now saying number 1 and 2. So this means this retrograde labeled cell is also positive for signal number 2, while all these others are just for number 1. And you can get a lot of data on this with these different tables here. And you can also get data across all of the images in a folder, because this will read the information here. And if you are happy with this now, you save your counts, and it says data saved to this counts folder, which is here, counts, and cell data, and then the name of the image .tif.csv, which contains the data from this quantification that you did. If we look at this in the notepad, we see that we have the X, Y position of every cell, and we also have the set position, and we have the category, which is category 1 for most cells, but 1, 2 for these 2 cells here. And this data is used to save and load the counts, and to analyze later in the Excel file and make 3D modeling as well. We will look at that. But that's basically the idea behind this. And now we could reset all of these counts. OK, and we could load the counts again. Click OK, and now these are loaded again. And you can also choose if you want to work at the 2-dimensional level, that you don't see any difference or represent any difference between the Z positions, or you could associate all your multipoints like this with slices. Like now you see that you actually have some representation of these multipoints in different slices, and you can do the same with the overlays, these circles on and off. And so you see here, if I now take this off, then it will just be in all the slices. So these are some options you have to choose to work in 3D or 2D. So let's work in 2D, and let's just say that these points here, they are positive for marker number 3 and 5 and 8, for example. Click the green, and then we clear these multipoints, and we see that we have now some special cells that are triple positive for something. You can now also use this set category, that you can not add new markers to an existing cell, but that you set the category. So for example, now if I want to set this cell that is 1 to 8, and also this one, these two, then I just have set category. I want to set this to 8. I click this, and as you see, these two cells are now number 8, category 8, and I save the counts, and then I have all this data with my cells. But this was just some clicking around. Now let's try to actually quantify this data. So we reset everything, we click OK, and we Now we want to use some automatic detection as well. So then I go to this panel and I put some blurring to the image for detection, because that has a tendency to look at these signals as a single signal and to center the signal as well. So my markers are placed in the middle of the cells, and I put the noise tolerance very low. Maybe we can try 5, because then I will also include these cells, because now I want to detect all of the magenta cells, and I want to detect any type of color, but I only want inside the outline of magenta color. So I only want to see what's inside here, and then I click find 2D maxima, and now it has tried to detect all these magenta signals. Here we see that it kind of did a mistake, so I will hold down Alt key and left click this, and then I remove one, and I'll change this position a little bit. And here you might also do some changes. You can go into the set stack and you see that yes, these are indeed 2 different cells, so I updated that. And now I say that these are all my retrogradely labeled cells. So I select category 1, I go to add category. In the initial step it doesn't matter when you don't have any overlays, and you click this. And now this is the first step. So I identified all my cells. Now I want to say which ones are positive for this LBX1 marker and which ones are positive for the HOXP1 marker. So I start with this, and I say again I want to detect only inside magenta, and I can detect any type of color because I only have one color here, but I increase my noise tolerance so that I don't detect this very dark signal. So let's try 50, see if that yes, it detected, but also this one. So if I have lots of images like this, I might look for a perfect value here. Let's try 65. Yes, that seemed to only detect the positive ones. And then I say this is marker number 2, and I click this and clear, and we can see that these have been identified as different cells than the others. And now I want to say which ones are positive also for HOXP1, so I change to category 3, and I use the same settings because it's very similar, and yes, it detected mostly correctly, but a few incorrect, so maybe it should be a bit higher here in this channel. Let's try 80. But the point is that this paper is called semi-automatic cell-by-cell analysis. So it's semi-automatic because you have some automatic capabilities here, but you can change it very easily by correcting the errors that you observe, and you can also compare with the verification channels, and you can get very reliable colocalization analysis. And let's convert this last multipoints to actual counts, and we clear the multipoints, and then we save our counts. Yes. A few things I can say more is that you have some other options here which can be useful. You can change the color of everything here also, and if you want to do 3D reconstructions, you can say that this is like the origo, and this is the angle of north, so you save this, and then you can also draw some contours representing the outline of your tissue in every image that you have, and you can make 3D reconstructions that we will see in the Matlab script that is provided. I think that's pretty much it. You can have a look around, play around with this. And lastly I want to say something about that this is a bit a subjective way of analyzing, because it's relying, yes, on some objective automatic detection, but there is also some room for subjective interference here, which is partly why we have this logging feature. So every click that you are doing is actually logged. Here, if I create new points manually, this is logged in this file, and if I convert this now, then it's logging this, and if I move anything, everything is logged, and this is automatically saved to this counts folder in the counting logs, which you see here. So if somebody, a journal, asks about this procedure, you can have this counting log showing what you have done for every step, and it's also logging the automatic detection. So if you try to detect, it will now log this, that you did a detection, it found 4 cells, and you converted that using category 3, etc. So that is a way to deal with this subjective part of semi-automatic quantification. Now let's look at the Excel file, which is this step of the toolbox where you can summarize the data of your quantification. So the plugin actually automatically creates this Excel file .xlsm in this counts folder. So the only thing you have to do is to go into the counts folder and open this file in Excel. I will do this now. And then you should enable macros, which is used, and you should press yes for this question here. And then you go through these 3 steps. First step is to check and validate the data, and it just looks at this folder, and looks at the cell data and the contours data that we drew, and sees if everything is okay. In this case we only have 1 image. And then you go to step 2, you can create some settings and options here, and you actually import the XYZ coordinates. It says everything is okay. And then finally you click the 3rd button, and that will summarize all the data. So here we have a summary, so it shows that this output image.tif, we had 27 cells positive for this one, etc., etc. And here you have calibration, contour data, actual cell data. So you can actually go into the individual cells here, and you can change the values. For example, if this cell is wrong, you can delete it, and then you can again export and summarize the data based on these changes that you did in the cell data tab. Let's for example go and delete all of this 1, 2, 3 category. So you see, if you remember, we had this here. Let's delete all of those, and then they should disappear from the summary. Yes, now we only have category 2, category 1 I mean. So this is quite useful, and I will now load up example data set which has data from 3 images in consecutive slice position throughout the brain. So this data that we will look at now is actually from example 3 in the paper, where we are working with large images, doing 3D reconstruction and some quantification. So we have here input 3 images, and we do the quantification, and then we do the Excel step, and then we visualize the data in Matlab in 3D, which will result in this kind of figures. So 3 sections, 3 images, do the quantification, and then let's look at the Excel step. So first we check and validate, and then we see that we get the 3 images here, image 1, 2 and 3, and these are of course separated by some set level, so we can manually input here the distance between the images in pixel value, and here is some calculation about how to go from micrometers to pixel value, or we can manually write it here. And we see that we have draw contours, tissue contours for all of these images. We have set the origin and the north, which means that we can actually do the 3D reconstruction. If you remember the origin and north, we set with this line indicating where is the middle of the picture in a 3-dimensional common reference frame. So for example, you can choose the midline of your tissue, a vein that is going through all of your images or something similar to indicate where they should be relative to each other. And then when you have done this information here, it's basically just this you have to worry about, and this. Then you do step 2, which is to actually import the data from this count cell data folder for each of the images, all the counts. And now that is done, and as you see, we have imported the cell data for image 1, for image 2 and for image 3. We have lots of different XYZ values, and we have some categories, differences 1, 2 and 3, 1, 2, etc. Only 1. And here you see the set slice position. This was analyzed in 2D within the individual set stack images, but you can also, as I told you before, add the additional slice information here, so that will be taken into account. And now for all this data, of course these will all have the same set value, but you can add some random set jitter to the values, so that not all of the objects here will be in the same set position. Then it's a bit easier to observe and inspect. You see that there are some random set difference here. Set is going up and down here, and you can choose to use or not use the set slice positions, and you can set the set slice interdistance between the individual slice set stack slices, and you can choose which is the first position of this set stack, and you can reverse the set order, so and invert X and invert Y. And once you have set these options, you click this button, and then it will use all of this information and all of the origin and north contour data and everything, and everything will be summarized in no, it will not be summarized, it will just be calculated here. And finally, once you click the third button, because now you can go and you can change values here if you want, as I showed you before, but now when you're happy with that, you click this export and summarize combined data. Then you will get the summary data, and you see the summary here. But what you also get is this export, so you get everything combined in a single file, which is this cell coordinates.csv, and this shows every file in a every cell in a global coordinate frame with the X, Y, and Z and category, and this is what will be used in the Matlab 3D model. So you can also define the colors of the Matlab 3D model, like which category should have which color. And here is the coordinates for the tissue contours. But you don't have to worry about these files. All you have to do is open this Matlab file, and we can see how that. So first of all, how to, if you don't have Matlab or you have never used Matlab, then you can go to this download and installation section of the paper and read this, and it will explain that there is a way to use Matlab for free. You can download it free from here, which is like not a full version of Matlab, but just a runtime version, which means that you can actually run some of the scripts, Matlab scripts. So you download the newest version here for your operating system, and then you can run Matlab scripts. And the Matlab scripts are automatically created when you click this button. Then it will summarize and everything, and it's written here that everything is now put into the export folder. So you go into the export folder of your accounts, and here you see two .m files, and these are the Matlab files. So you would open this in Matlab, and there is one with a graphical user interface and none without one version without that graphical user interface. And I think maybe the one without works best for in the free version. But let's see, now here is a full Matlab installation. So let's just open this graphical version. So now we have this Matlab script file in the same folder, in the export folder, where we also have this all the data that we need. So we run the script. Let me check if I'm recording. Yes. So I open the script here. This is a simple script, and I click run, and it should look for this data automatically. Here you can, it's asking where is this data, but it should be okay by default. You can set some transparency option for the tissue contour, and some different values here, so, but there's not a lot of options. You click visualize, and now you see the data that you have been analyzing in your ImageJ in 3D, and you can rotate this around, and you can inspect the different groups of cells in different colors and see if you see some patterns of clustering, etc. This is now running a bit slow on my computer because I'm doing this through a virtual machine, and now it's a bit better. So the tissue contour is not looking very nice, so let's try to, here it's information about the colors, what they represent. You can try to change this alpha value, determines the level of detail. Smaller values, more detail, but too small yields no shape, so I think we should increase this a little bit, 900. And now that shape is looking a little bit better. Yeah, so this is the tissue contour, and these are the cells, and you can inspect this in 3D, so you can make reconstructions of all kinds of colocalization data. And this is what I used in my other paper, Lunde Okati et al. So if we go and have a look at this, I'm not sure if this is yes, this is open source. Here you can see some figures that were made with this procedure. So we are doing molecular profiling in the brainstem, looking at a lot of transcription factors of some neurons, and we have plenty of figures. Maybe we should go to this tab. So just so you can see an example of what kind of figures you can create with this tool. Here you see the brainstem with some cells. Here you see different categories, similar idea. Here you see brainstem again with many different categories of cells and their distribution in 3D. So this is of course recreated from many individual images, where we have drawn the outlines on each image and indicated, quantified the colocalization categories. So hopefully this is helpful to you, and please reach out to me or provide any feedback in the comments if you have any. Thank you for watching and good luck with your work.